chơi game mà cô?
Oh no. Quên ta. Hôm qua.
Alright, students, we start the lecture today by playing the game to revise the lesson from the previous lecture, okay? that cannot join the games? I see that's just only 183 students here. So for the red student, I think in this auditorium there is more than 200, okay? So why there is only less than 200 students join the games? It's just one time for you to practice the lesson, the previous lesson, and I think it's one time for you to practice before the exam, because the question that's up here in the games is much similar to the question in our exams. I will start when we get 200, okay? Now please indicate your student ID instead of your names or your nicknames, because if you have the best result in the top 10, then we receive the bonus point. So let's start to revise the knowledge for previous lessons. This ranks as the easiest question in your exams, and in this question I hope that the whole class will have the right answers, but only half of you need the right answers. So the substance acts as a medium for biochemical reaction in the cell. We have the water, so oligomers or oligomers reaction inside a cell happens in aqueous environments. So the water is choose correct answers. because it simply defined that the carbon glucose contain carbon, hydrogen, and oxygen atoms. And even through all of the above, for example, if this pentyl sugar is not quite correct because you know that we have various types of sugar. And is it polysaccharide? It's not true because carbohydrates, glucose is widely fused and it contains monosaccharide, disaccharides, or polysaccharide is not only polysaccharide. So for these questions, the correct answer is C, it contains carbon, hydrogen, and oxygen.
Kalau yang naik protein di
question, right? In the covalent bond, electrons are shared, and in ionic bond, electrons are gained and lost. Give and receive, or some words are similar meanings. May I ask the students, BI 2243? BI 2243, you're here? Yes, please. Can you explain the answer of these questions?
Okay, so I understand about it's like a connection to and it's like a bone of the cell to have to like save it because it have a connection. I don't really know how to explain it, but it's like the bone of the cell.
It's like a bond of the cell, so why you choose these answers?
Yeah, it has a
This strong bond is the bond of the cells.
no, it's not the really strong alone. It can be broken.
So why you cannot find the hydrogen bond between the side of two DNA member helix?
So I can't really because it's a definition of the hydrogen bond.
Okay, stay there. You know go to the next questions and we'll come back with you later.
protein.
I turn it to spot you can see that. So, okay, analysis is the correct answer. In the final exam, I just see some students, I think just 1 or 25% they have the wrong answer of this question. I don't understand why, but for today's I'm satisfied with these results. So the basic units of protein, we have the analysis. A lot of students will do the amines, but the amine is just one group of the analysis. We have the one carboxyl group, one amine group, one A group connects with the alpha carbon in the center and it's all form an amino acid. Move to the next question. Okay, the very small amounts in our bodies among these elements, yes, it is calcium. So we have carbon, hydrogens, and oxygens and nitrogens occupies 96% element inside our bodies. And if we account for more phosphates and sulfur is occupies 99%, and calcium is very small amount. So the secondary structure of proteins. The first answers the linear sequence of amino acids linked together by the peptide bonds. It is the width level structure of proteins.
So the first structure of protein is the linear sequence of amino acid, which is linked by the peptide bond between every amino acid. And the second, the second is that the linear sequence, it either falls into the alpha structure or it's just kind of like become the no, I'm sorry. Either it's kind of a to alpha or fall into a beta sheet. Yeah.
Okay, for the rest.
For the rest, the answer of various loops and terms, I don't think that is the secondary structure. Loops and term maybe, or maybe the third structure, and the answer of at least 2 amino acids is the fourth structure, because between from the first structure to the third structure, it can only have one amino acid. So that an option is not really.
Which structure has just only one amino acid?
I think so.
In the chains of amino acids, it's just only one amino acid. We cannot call it the proteins.
But yeah.
So you're right. So the first, pay attention, because the linear sequence of amino acids, they are the first level structure of proteins. The second, whenever it appear in the exam, if we have the alpha helix or the beta sheets, mean the secondary structures. And whenever you see loop and turns, remember about the tertiary structure of proteins. And for the rest, when you see 2 chains of amino acids, more than 2 chains of amino acids, mean the quaternary structures. I just focus because it's difficult for you to remember the terms and the fourth structure of protein, so I emphasize here the words loop and turns for the terms, and at least 2 chains of amino acids for the quaternary structure or the fourth structure levels proteins. If you're really not interested in my lectures, just focus on the keywords that we will have you to pass, okay? this question, you will choose more than one answers. So basically, most of the enzymes are made of proteins. So the pH and temperatures do affect the activity of an enzyme because you know that if we change the pH or we increase or decrease temperatures, it may affect the protein, especially when you increase the temperature is too high, your protein will be denaturate. And for alkalis, it's some words. That is not quite precise in this case. For alkalis, it's just a part of the pH, you know. Alkalis is two bases and acidic is mean too much pH. The pH is quite low, right? Alkalis pH is quite high. So it's not quite specific for this case. For the rest, the substrate concentration, yes, they do affect the activity of the enzymes. It's too much substrates, but when you have two liter amounts of the enzyme, this may slow down the activity rate, and in the reverse case also. So our correct order is two aminoacids can combine together to form a dipeptide by a covalent reaction, or this is how many students have the correct answers. For these results, this means that at least more than 100 students have the correct answers, right? I think there's a problem when I define the point here. Okay, we move to the next question. do it in the computer, so you have all these multiple choice questions. Sometimes it's matching questions. Sometimes it's insert the words and try to insert the precise words because I'm not the one to evaluate the test. It's just computers. And also another type of question is reordering question. Like the previous one, you just have to do the reorders questions. And we will see to the finals.
Final session. Rồi còn giữa cao cũng được.
Who have the at least 70% corrections? 70%?
60 đó.
Yes, at least 70%.
Thêm phần tỷ.
More than 70%. What can I do? Because I think that if you have achieved at least 70% correct in this test, you will pass my exam. But I think not a lot of students achieve that score. So please practice more, and in the tutor class, I will try to give you the more exercise to practice at home. And this year, I expect and I will try to have 80 to 90% of students pass my course. And I cannot do it alone, so please practice at home. So 0.5 will give to the top 10 students today. So we experience together two parts of my lectures. The first part is interview about the cells, the domain of life, and the second we study about the chemical bonds and the macro and small molecule and macromolecule inside cells. And today we will go further deep inside the cells, how things inside the cell can contract the cells. In eukaryotic cells, we study all about the eukaryotic cell until we reach the lesson number 10. Lesson number 10, we have the lesson that we talk about the prokaryotic cells. So in eukaryotic cells, all the eukaryotic cells contain the three basic cell structures. The first is cell membranes, like we do have the skin to protect our bodies, and distinguish our body with the environments, and the cell also, they do have the cell membrane. And the second very important part is the nucleus. It's like, we can say that it's like our brains or is our nervous system, which contains all the information and controls all of the reactions inside the cells. And the last part is the cytoplasm organelles. Cytoplasm here is the thing that's inside the cell membranes is like our blood, okay? Cytoplasm with organelles is like our blood with our organs. So move to the first part. What about the cell membrane or the skin of the cells? It's composed of double layer phospholipids and the proteins. Remember the phospholipids in the previous lessons? Yeah, the phospholipids, two parts, a head and a tail, right? So the plasma membrane is contained of phospholipids. Why we call the double layer? Because double layers need two layers of phospholipids to make the cell membrane, and also the proteins. So it's surround outside the cells, like the skin is surround outside our bodies. And they have a specific system, proteinaceous complexes, for transporting iron and other solute into or out of the cells. It is living layers because have a system for signaling between the interior of the cells and the external environments. It's like our skin, you can feel or you can feel and receive the information from the environments, like where you can send the temperatures. It's not really like the cell membrane here. Like if you want to send or to transport the signal between body and bodies, you need to speak, you have the eye contact, you have several ways, another ways, like it's not like our skin here for these informations. So the plasma membrane structures is composed of the phospholipids. It is two parts. The head is the glycerol and the phosphate group, and the tails is the fatty acids. And the head is the hydrophilics, attract the water, love waters, and they have the tails is the headwaters, hydrophobics. So they can make up a bilayers. Bilayers is double layers or two layers. Where the tails here, you can see the tail hairs, they point inward towards and make towards each others, and the head is turned out of the membrane. So it's a living layer, so it can and the bilayers can move laterally to allow the small molecules, oxygen, carbohydrates, and water to enter and out. Some bigger molecules can go through the protein channels here or for the water because they are polar. They have a specific aquatic channels. So we will move. We can go deeper about the cell membranes. We have one lecture about cell membranes, so I will go very fast for this part. Cell membrane proteins also we will discover in the lecture number 4, I think. And pay attention about cell membrane of plants. They lie immediately against the cell walls. In the plant, they have a cell wall surrounding them. Remember about the first man who discovered the cells about the Lowen Hook, when he saw cut layers of the plant under the microscope, he just see these small rooms And this kind of small leaves made up the cell wall of the plant. So here the cell membrane of the plant is again the cell wall, and it push out again the cell wall to maintain the cell shape. And we talk about the cell walls of the plant is found outside of the membranes. They are non-living layers. Its important function is the cells include protection, structure, and support. And we can find the cell walls not only in plants, but also in fungi and bacteria. Remember this important. For the cell wall of the plants, it is made of polysaccharides or the cellulose. In the previous lesson, I talked to you. Plants, polysaccharides. Fungi, the cell wall is made of chitins. Bacteria is made of peptidoglycans. And the archaeas is made of various types of compound glycoproteins, the S layer, the pseudopeptidoglycans. Pseudo means that fake peptidoglycans are polysaccharides. I think the question about the cell wall is most appealing of exam is focused on the structure of the cell wall only. So we move to the second important part. We have the nucleus. I will go very fast because we have another lesson about the nucleus. So the nucleus contains the genetic materials and is the largest organelle in the cell. Largest organelle is most with the is not really true with the plant cells, because sometimes in plant cell or in the most plant cells, the biggest organelle is the vacuole. And here, so in general case, the nucleus is the largest organelle in the cells. It's bound by an envelope consisting of double membrane, and the genetic materials are concentrated in a part of the nucleus. Nucleopores provide the means for transport across the envelope for large molecules to enter or leave the nucleus. It seems not sometimes difficult to the student who's not focused on the biologies, but don't really have another lesson about this. I'm going too fast about this part. So in the nucleus, the genetic material is found. We have the chromatins is appeared in the non-dividing cell, and chromosomes is condensed in the wrap around the protein form the chromosome in dividing cells. For the DNA, it's the hereditary material in the cell, and genes, also a gene here is the genes. Talk about the genes, in the basic physical and functional unit of heredities. The genes made up DNA molecule code for different proteins, but some genes do not code for the proteins. Some place or some gene is it's just we just need the RNAs from them, and some microRNAs also, and they do a regulatory growth. It's not only the code for the protein. Inside the nucleus, we have a small part we call the nucleolus. Here, then gone. So it is inside the nucleus. They will disappear when the cell divides, and they are the signs of ribosome biogenesis. So inside the nucleus, just remember for me, just small parts called the nucleolus, and the ribosome synthesize from that part. So we move to the next part, it is ribosomes. The ribosomes here is the protein factories for the cells. It is a site of protein synthesis. They float in the cytosol and they can make proteins used inside the cell, or you can stick on another part, another structure inside the cell. We call it ER. We will discover it in the next slide. So the ribosome stick on the ER will be used inside, and also most of the protein to export it will synthesize in the ER. Here in the board is cytosomes. I think I have some mistake if I don't explain to you. We have cytosomes. In the previous slide, we have cytoplasm, right? Do you know what difference between the cytosol and the cytoplasm? I think here you will see some word. We have cytoplasm. We have also protoplasm. And the rest we have a cytosol, right? So here the protoplasms is everything inside the cells is contains cytoplasm plus the nucleoplasm. So thing that's inside the nucleus here, we have some things. It's quite like the cytoplasm cells. So the cytoplasm plus the nucleoplasm, we have a protoplasm. And the second part, cytoplasm, is everything inside the cell membranes, but exclude the nucleoplasm. Okay? And for the last, cytosomes is the things in the cytoplasm minus the organelles. Okay, we have two terms here. So cytoplasm is just the like your blood, right? No, cytoplasm is blood plus organs inside bodies. Cytosol is like your blood. Protoplasm is blood plus organ plus things inside brain. Okay. So here's ribosome may float into the cytosomes. Sometimes some word here, if I don't explain this, maybe you don't understand what difference. About the ribosomes in the site of protein synthesis, so have anybody here vaccinated with the Pfizer vaccine?
Vaccine here.
Pfizer.
Pfizer, vaccine COVID của hãng Pfizer.
Yes. Yes. So most of you have been vaccinated by AstraZeneca.
Me, Pfizer.
So do you know what is Pfizer and how it works inside your bodies? How it works inside your cells? Yes, please.
Okay. Okay.
Hello.
Okay, so I here some explain about the doctor the difference between COVID-19 vaccine AstraZeneca and the COVID-19 vaccine.
No, just ask only about the Pfizer.
Okay. The vaccine Pfizer is contain a little bit of COVID-19 virus and
COVID-19 virus. not kháng thể.
Không phải ý nó là con là tiếng Việt cho nó dễ hiểu, đó là sinh.
Nói
Thôi tiếng Việt thôi, bởi vì con không của tiếng Anh trên lắm thì bên trong của cái vaccine COVID-19 của hãng Pfizer thì nó sẽ có một chút cái con vi khuẩn, một chút con virus COVID-19 đấy, và thành phần và thêm cả kháng thể để đã khỏi bệnh.
thêm kháng thể, virus của kháng thể trong Pfizer để tiêm vào cho mình khỏi bệnh. Đứa nào vỗ tay? Đứa nào vỗ tay? Who just clap your hand? Please explain this question for me. Please give the class some explanations.
Em thấy là bạn Lâm nói hoàn toàn chính xác rồi.
So you totally agree with me. You're excited and you clap your hands. Who have other explanations?
Astra như là chỉ có cái gì ạ?
What is Pfizer? So why when I talk about the ribosome, I asked you about the Pfizer?
Họ có Pfizer lạnh Astra.
Okay, so about the Pfizer. So why we talk about the Pfizer here in the ribosome part? So you know the ribosome is the proteins factories for the cells. It is science for the protein synthesis. Here you can see the mRNA. So other name of Pfizer we have is this one Pfizer mRNA vaccine. Ôi, vaccine RNA, RNA, đúng không? Vaccine RNA. So when you inject the Pfizer, it means you inject the mRNAs inside your bodies. So what these mRNAs is bringing information for? Cả lớp này không đứa nào tiêm Pfizer à?
Đây em tiêm đây ba.
Tiêm không biết tiêm cái gì mọi người. Không có biết luôn. So when you inject the Pfizer vaccine, this means you inject this part, this small part, the mRNAs. It's called the messenger RNAs. RNA thông tin là nó mang thông tin cho gì nhỉ? Cho cơ thể cái đầu mày đi. mRNAs bring the message, we call the message, so it's bring the information for for ribosome inside our bodies, and our ribosome will read this message and synthesize proteins. Do you know which proteins? It is spike proteins of the virus. So when the strange proteins appears inside your bodies, your immune system will work. And next time when our immune system see the real virus, we have the ability to find them, okay? Thôi nói lại tiếng Việt cho bọn không biết tiêu cái gì mọi người nhé. Nào mình tiêu Pfizer, thằng kia bảo Pfizer chứa virus và chứa kháng thể là hoàn toàn sai ạ. Em biết mà em vẫn phát biểu. Rồi mình tiêm mRNA là mình tiêm mRNA là cái thông tin của con virus mọi người ạ. Về mình tiêm vào bắp đúng không ạ? Sau khi mình tiêm vào thì cái mRNA được đóng gói trong một cái lớp màng nano lipid particle. Cái này bọn nào học nano nên mày sẽ biết là mình đóng vào cái màng như thế nào. Cái màng này sẽ fuse vào mình tiêm bắp đúng không ạ? Nó sẽ fuse vào cái membrane của cái tế bào chỗ bị tiêm đúng không ạ? Và nó sẽ giải phóng really the mRNA inside our cells. Tôi ta là tiếng Việt hết nhé. Mình tiêm mRNA đúng không? Nó bọc trong một lớp màng này nó sẽ fuse, nó sẽ tiếp hợp với cái màng ở trong tế bào trong cơ thể mình và nó sẽ giải phóng cái mRNA này vào trong cái tế bào chất của trong tế bào đúng không ạ? Và cơ thể mình sẽ dùng chính ribosome của cơ thể sẽ đọc cái thông tin này, đọc cái mRNA này. Đọc chính là tổng hợp protein ấy, và nó sẽ tổng hợp lên một là protein, đó là protein vỏ của con virus ạ, spike proteins. Sau khi đọc thì nó sẽ gửi cái protein vỏ này lên cái bề mặt tế bào chỗ bị tiêm ấy. Bề mặt tế bào nó tiếp nhận được cái mRNA này, và như thế cái tế bào đấy nó sẽ có protein của virus trên bề mặt và nó là protein không phải trong cơ thể đúng không ạ? Nó là protein lạ. Vì như vậy thì cái hệ miễn dịch của mình sẽ nhận ra là có một thằng lạ, thằng stranger trong cơ thể, thì tế bào miễn dịch, hệ miễn dịch của cơ thể mình sẽ đánh nhau với cái tế bào giả đó, đánh nhau với cái tế bào mà có spike protein trên bề mặt đó, protein của virus. Và sau khi đánh nhau thì nó sẽ làm sao ạ? Nó sẽ nhớ. Mình có một cái cơ chế hệ miễn dịch là mình sẽ nhớ, và lần sau khi mà cơ thể mình bị con virus nó tấn công ấy, thì bằng cái hệ nhớ của hệ miễn dịch trong cơ thể thì mình có thể đánh nhau với con virus hiệu quả hơn. Tức là khi đó mình đã có thông tin một phát mình nhân lên hàng triệu tế bào miễn dịch trong cơ thể và có thể diệt virus nhanh hơn. Đó là cơ chế của mRNA vaccine. Còn AstraZeneca is another case, and maybe I will talk about it in another part. So ribosomes, a very important part, synthesize the science of protein synthesis is translate the message from the mRNAs into proteins. So for ribosomes, this can be found in both prokaryote and eukaryotic cells. We have in prokaryotic ribosome is made of two small parts. Are 50S and 20S, and together they form a structure 70S. And for eukaryotics, we have the two small parts, 60 and 40, and together they make 80S. So, questions? What we can take advantage of these differences? Both prokaryote and eukaryotic cells do have the ribosomes, but the difference in the structures, 70S and 80S. What can we take advantage of this difference? là cả tổ tư vấn không tư vấn được rồi. Bất lực.
Cô nhắc lại câu hỏi của chị.
Các bạn học tiếng Anh thì các bạn hiểu Việt.
Sao cho là dễ hiểu.
Rồi, cái ribosome này mình có thể tìm thấy ở cả tế bào nhân sơ và nhân thực đúng không ạ? Cấu trúc của nhân sơ và nhân thực khác nhau. Ở tế bào nhân sơ mình có hai tiểu phần 50S cộng 30S và tạo nên cái 70S. Trong khi đó ở eukaryotic cell là 60S cộng 40S thì tạo nên 80S. Câu hỏi của cô là mình có thể dựa trên những cái khác biệt giữa ribosome của prokaryotic cell và eukaryotic cell mình có thể gọi là gì nhỉ? Có lợi ích gì hoặc là mình dựa vào cái sự khác biệt đó mình tạo nên lợi dụng sự khác biệt này mình có thể làm gì nhỉ? Now 0.5 for the ones who can find the answers. Any ideas? Là gì? Google. Google là gì? Tắt phải bắt.
Nó lại từ cô con thích cái gì? Okay, the prokaryotic ribosomes are the smaller than TS particles, while eukaryotic ribosomes are larger, ATS particle. Okay, this is the key difference between the ribosome of prokaryotic and eukaryotic. Furthermore, another difference between this is ok. Thế thì Google lại phát. Ý cô hỏi là cái mình có thể là
Cô hỏi bài bằng tiếng Việt ở đấy.
là lợi dụng được cái sự khác nhau của sinh vật nhân sơ và sinh vật nhân chuẩn thì nó có cái tác dụng gì đúng không? Ok, cô đợi em xin.
Câu hỏi rất là mệt. Sân Google bằng thế. So you know that the ribosomes is the site of protein synthesis, so they do important functions in both prokaryotic and eukaryotic cells. And we can take advantage of these differences by produce the antibiotics, targetings on the ribosome. So when we take some drugs, some pure drugs, so we hope that it could be some selective effects, right? It will kill the pathogens, to kill the bacteria instead of our cells. So we can create some drugs, some antibiotics with targetings in the 70S ribosomes of the prokaryotic cells, but not ATS of eukaryotic cells. And generally now is more than 50% of antibiotics is targetings in the ribosome. Okay. Whenever we found the difference between the prokaryotics and eukaryotics, we can take advantage to create the drug. And the next part we're going to discover together is cytoskeletons. Cytoskeletons, we have another lesson about the cytoskeletons, and I go very fast. This picture is more clear for you to see the cytoskeletons here, the green part. So many green parts here, you can see the cytoskeleton. So you do have your skeletons and the cell also. It's the same function, that's maintain the cell, maintain the shape and helping you in the movements or something, okay? This is the cytoskeleton. The pictures when we're staining the structure inside the cytoskeletons is present in the cytoplasms of all the cells, including the bacteria and archaea. They have maintained the cell shape. Pay attention, the cytoplasms of the bacteria and archaea is quite different from the cytoskeleton of the eukaryotic cells. They contain some proteins that compose the cytoskeleton is similar to the protein that compose the cytoskeletons of in the eukaryotic cells. The function of cytoskeletons is to maintain the cell shape and the movement of the vesicle and the organelle within the cells. And they are made of proteins. We have the microfilaments and the microtubular. This is the microtubular, and the small part here, the small tube here, we call the microfilaments. We will go deeper inside this cytoskeleton part in next lesson, okay? Alright. So I think we have 20 minutes break.
Không bu thì
So as we continue lessons, keep quiet please. We move to the next organelles inside cells. It is endoplasmic reticulum, or how do we call it in Vietnamese? Okay, mạng lưới nội chất, endoplasmic reticulums. And this is the network of hollow membrane tubular. Nó là mạng lưới ống rỗng ạ. Connects to the nuclear envelope and the cell membrane. And the function synthesis of the cell product and transport. We have two kinds of the endoplasmic reticulums. Is the abbreviation of it is the ER. So two kinds of ERs. We have the rough ER and the smooth ER. You will see they are completely different here. Mạng lưới nội chân, chân và smooth ER, and mạng lưới nội chân hạt we have rough ER. This is the rough ERs. The rough ER, they have the ribosomes on its surface. And because they have the ribosome is the site of protein synthesis, so the function of the rough ER is also make the membrane proteins and the proteins for the export out of the cells. So the rough ER, they have also the function is makes proteins. So here's in the surface of the rough ERs, they stick with some ribosomes, and then when the protein synthesis in the surface of the rough ER, and then this goes to the interior periods of the rough ER to be modified and transformed. The other types of ER, we have a smooth ER. So here in the center here you can see nucleus here, and then the rough ER is surrounding them, and then we have smooth ER via this picture. Smooth ER, lưới nội chất trơn. The functions of the smooth ER is to make the membrane lipase, or we have a steroid is made here. Sometimes it can regulate the calcium, especially in the mast cells, and it can destroy the toxic substance. So, which cells in the body do you think that have big part or have a big smooth ER? Which cells will have the more smooth ERs in the cell, in the body? Thận. Why thận? This is more smooth ER. Like the macrophage. Why macrophages have more smooth ER than the other cells? Tổ tư vấn cho em này là cái gì?
So in my opinion, the macrophage, it has the role of làm nhiệm vụ bảo vệ cơ thể. So it has to export all toxic substance, and therefore the need for more smooth ER is necessary because it has to fulfill the role of destroying kiểu như là trung hòa độc tố ấy, trong cơ thể của tế bào, bởi vì cái mạch cầu đấy nó làm việc nhiều và nó phải hấp thu các chất độc từ các cơ thể, hấp thu chất độc từ cơ thể, nó phải giải phóng chất độc.
So you mean that the macrophage you neutralize the toxic compounds?
Yes,
And
also you have
eliminate the toxic compound.
huh?
Eliminate.
Yes, eliminate the toxic compound.
I think the purpose of the macrophage is to find the bacteria, virus, and eat them.
Yeah, because they eat them, so there are certain.
But they are not a toxic compound.
But
So-
they are strange to the body, so
Yeah, they're strange.
so they might be able to toxicate the cell, so it has to develop some way to resist.
I think about the detoxification organ inside your bodies. This could be the detoxification organ inside your body.
Well, the most toxic part in my body, I think that could be the liver.
It's not the most toxic part. It's the part where detoxification process happens inside our body.
Yeah.
Gan không phải là bộ phận độc nhất cơ thể nhé. So inside our body there is one organ that plays important roles to detoxify the toxic compounds. And also it's not only the toxic compounds, sometimes when you take some drugs, the very important process will happen in the liver. And you see that the function of the smooth ER is destroy the toxic substances. It means that the liver cells need to have abundance of the smooth ER. So when you take some drugs, most of the cases in the insoluble form, and the function of the smooth ER is to add the hydrogen group to the drugs or either the toxic compounds, and then make them soluble inside the bodies and easy to eliminate outside of our bodies. In the same way when you take some wine, the ethanol will move to the liver cells, and by the smooth ER inside the liver cells, it can transfer the ethanol into the acetate. So it can and they detoxify the ethanol. The next part inside the bodies is, I want to introduce the Golgi body. The Golgi bodies is the flattened sap, and they have a two phase, a seed phase, or we call the receiving phase. This phase is receive the vesicle from the other organelles, and a trans phase we call the seed inside a trans phase. So it's received in the seed and then relieved in the trans phase. There are two phases. So proteins will be received in made of ER, then received by the seed phase. As the Golgi bodies will packaging all these compounds and make them more mature. I can say that they make them more mature, and then transfer them in the trans phase. With the trans phase, they get fused to the membrane for relief from the cells. This is the transport vesicle. Vesicle các bạn có thể hiểu là những túi nhỏ. So they receive the vesicle in the seed phase, fuse membranes, relieve the compound in the Golgi bodies. In Golgi bodies, they will package them, make them more mature, and then send them in the trans phase. We have here the graph that represents briefly how the proteins can transport between endoplasmic reticulums and the Golgi bodies. So here in endoplasmic reticulum, they can clot the proteins by membranes and by buddings. Protein will be caught in the membranes, buddings, and into here in the vesicle, small vesicle, and transfers to the seed phase of the Golgi bodies, and for the rest is transferred. So we receive the vesicles by the seed phase. This budding, this vesicle will fuse into the seed phase of the Golgi bodies, and then is released in the trans phase. This is the way how the cells can package in some proteins of some compound and send them from this organelle to other organelles. Our next part we experience another organelles inside the bodies is the lysosome. The lysosome actually they are specialized vesicle that holds enzyme inside. So they hold the digestive enzymes inside. These enzyme proteins various ways were first created. The lysosome can break down the food, bacteria, and root out the cell parts of the cells. And also the inverse in the program for the cell mates, or we call the apoptosis, we experience in the lesson number 10. And also they can lyse and release enzyme to break down and recycle the cell part. So do you know that the proteins are the enzymes here, and in the lysosome ways for create. So the enzyme is proteins, right? So can you guess where this enzyme the first creates inside the cell? the source of the enzyme inside the lysosome? Lysosome is the package of the enzyme inside, yes.
I think because the lysosome itself is the package, so so it means that it must have been created inside the rough endoplasmic reticulum. Because after it is created, it has to be moved to the Golgi to create the package so that all the enzymes are packed in one package.
Sorry, another question. Can you guess which cell types inside our body have abundance lysosomes?
I think it could be the bạch cầu.
Đam mê bạch cầu.
Because as we have said before, the macrophage,
Yes,
macrophage has to digest all of these strange pathogens, and they have to eat all the pathogens and all the strange substances in their body. So therefore, it has to have lysosome, in abundance of lysosome to actually digest that amount of bacteria, pathogens, and substance.
Okay. Okay, so 0.25 for you. You answered a lot of questions. So the lysosome is like our stomachs. We can consider they are the stomachs because they have contained so many enzymes inside. And the function of the lysosome is to digest some things. It could be the pathogens like you said, maybe the bacteria, maybe the virus, and sometimes it's a small piece of the cells inside our bodies will be digested by the macrophages, and therefore the macrophage must have the giant stomachs. So this abundance of lysosome will appear in the macrophages to serve these functions. So we have another type of the enzyme package. We have a peroxisome. Peroxisome is small vesicles found around cells. Single membranes that contain the digestive system, digestive enzymes, and they absorb nutrition that the cell acquires, digesting fatty acids, alcohols, cholesterol synthesized, and digesting amino acids. Who knows the difference between the lysosome and the peroxisomes? The lysosomes is the enzyme package, and the peroxisome is also the enzyme package inside cells. Why we have two types of enzyme package? Two enzyme packages, and is also the digestive enzymes, the same. So why lysosomes and why peroxisomes?
I would like to say that, as you said before, the lysosomes contains the enzyme that destroys the particles, mostly to digest contains the enzyme to
Digest also.
no, it destroys the particles that are toxic, so it helps protect the body while the other ones help digest, so the body gets the nutrition and support. And the perisome here, the site is extremely small, but the other one is large.
One small, one big. So the size of this package is here. You just searching by Google.
No, you just said that, I remember.
So you see small vessel. Any difference? I'm sorry.
The Google said that another difference. The process of lysosome functioning, it does not release any energy, but in perisome we can release some ATP.
Release some ATPs. Peroxisome can release ATP. Okay, so one thing that I emphasize here is oxidative enzymes. So the peroxisome that they contains, the difference between the lysosome and peroxisome is peroxisome they contain the oxidative enzyme. That's enzymes requires oxygen to work. While in lysosomes, they are working in oxygen poor area. That they don't need our oxygens. And our cells want important information that is the pH. The pH of lysosome is quite low. This is two differences between the peroxisome and the lysosome. It's based on the enzyme that they contains. And can you guess the enzymes in the peroxisome is where they come from? So in the lysosomes, as the guns have, or just say that the proteins from lysosomes is come from the rough ER, okay, the package in the small vesicles, same the antibodies, the majors can work, and we have the lysosomes. So what about the peroxisomes? The source of enzyme from the peroxisome, where it come from? If they come from the different sources, Can you get the source? One come from the rock ER, enzyme from lysosome is come from rock ER, and enzyme from peroxisome, where it could be, where it could come from. Where is the size of protein synthesis inside cells? Now the size of protein synthesis is where? Cytoplasm? Alright, it's a bigger area. More specific? Of course, all the reactions, all the mechanisms inside the cell happened in the cytoplasm. For more precise, which is where is the size of protein synthesis? Which structure, which part of the cells have that role? We have the studies Trời ơi, các bạn này vừa học cái gì? Là nơi sản xuất protein nhỉ? Cái gì tham gia vào quá trình tổng hợp protein? Yes. The size of protein synthesis inside cells, we have the ribosomes. So the ribosomes, where we can find the ribosome inside our body, inside the cells. Where can we find them? One stick in the rock ER, lưỡi chân hạt, okay?
I think it can be found in some stick to the rock ER and some just float freely in the cytoplasm.
Yes. And can you guess the source of enzyme forming the peroxisomes? So can you get the source of the enzymes in peroxisomes? Nguồn gốc của enzyme từ peroxisome ở đâu?
I think that those ribosomes will produce lysosome, and then the lysosome will produce that enzyme that in the peroxisome.
Cái thằng này đã hỏi như thế rồi. Yes, ribosome is sure. But we have two types of ribosomes. First, this ribosome is stick in the root ER, and as Gerda says, the source of the enzyme from the lysosome is come from the root ER. And as I said, the source of the enzyme in the peroxisome is quite different because it creates from the ribosome that's free in the cells. We have some ribosome that's free in the cytoplasm. They don't stick to any organelles, so they can synthesize protein also. And the source of enzyme from peroxisome is come from that free ribosome. Okay, cái này nó định cho điều nó đến đấy nó sai. So 3 things. Lysosome is oxidants, pores, areas. They don't need oxidants, and this enzyme inside the lysosome is working at a very low pH. And the source of enzyme from the lysosome is come from the root ERs. Why is peroxisome? They contain also the digestive enzyme, but the oxidative enzyme that's require the oxygen to work, and not a very low pH, maybe the pH in the nature or neutral pH, and also the source of peroxisome is believed that it's come from the free ribosome in the cytoplasm. That is 3 things different between them. And also, as I remember, there's some student that asked me about the endosomes, right? I think I don't know if it's in this class or another class. I will not teach the endosome in these lectures, but you can imagine that our cell can eat some particles. We have the endocytosis, and then it makes the small vesicles with also the receptor. It is the early endosomes, and then inside that endosome is lowering the pH and send the particles in the late endosome into the lysosome. When the pH inside the endosome is lowering, we call the late endosomes, and then endosome is also filled with the Golgi, filled with the lysosomes also. This is about the endosome, and I think by one Google search you can discover so many information about the endosome, so that's why I do not reply that kind of question. And also I do not talk about the endosomes in my slide also, so no reply for these questions. If someone sitting here asking the question, please understand. We move to another part of the cells. Some cells have a part called the cilia and flagella. Just think that it could be the tails of the cell, okay? Sometimes the tails of the cell, feather the skin on the tails. So here's in the picture the cilia, and here the flagella. For cilia and flagella is function in moving of the cells, moving a fluid or a small particle across the cell surface. And cilia shorter is more numerous, and flagella is longer and fewer. Remember for me these 2 information, this may appear in your exams. This is one example of the cilia, or is moving away away the dust particle from the lungs. There's numerous of the small cilia here. It's work in one way, and all the dust inside your lung will be pushed out in your nose. Okay? The next part is the mitochondria, in Vietnamese, DP. So the mitochondria, remember, energy factories. Ribosome is the site of protein synthesis, and mitochondria is energy factories. All the living cells have a means of converting energy supplied by the environment into the common intermediates of ATP. ATP is adenosine triphosphate, eat the energy monies, the cells, and this is the structures of the mitochondria. The mitochondria is contains it has a double membranes. The outer membrane is smooth, and the inner membranes they fold so many times to increase the surface, and they form the structure called the crites. And inside the mitochondria, we have the fluids. We call the matrix. So the functions of mitochondria, they are energy factory. Yes, they convert the energy stored in food into the usable for work. Or this is the ATPs, and we call this process cellular respirations. So in the previous lessons, when I talk about the energy source of the cells, talk about the glucose. You know that glucose can be broken down into smaller molecule, and in each step, when we break down this glucose, it will relieve the energies in the form of the ATPs. So we can use the ATP to inside cells. Mitochondria is also have the let me come back here. The mitochondria, they contain their own ribosome and their own mitochondrial DNA. So if the question is if the mitochondrial genome is still functional. So the mitochondria, they are very special because they have their own ribosome and their own DNA floating in the battery. And it has been conserved during the evolution, and it retains similarity to the prokaryotic ancestor. So can you get the origin of mitochondria? So because they are similar to prokaryotic cells, there is a theory, we can get that the mitochondria is originated from prokaryotic cells. So mitochondria are thought to have originated from the Asian symbiosis that result when the nucleated cell engulf in aerobic prokaryote. Symbiosis cộng sinh Asian cổ xưa. Người ta cho rằng là mitochondria thì có nguồn gốc từ một sự cộng sinh cổ xưa giữa một tế bào có nhân đã làm tiêu biến một prokaryotic aerobic prokaryote, một prokaryote hiếu khí khác. And here is the this graph is represent these theories. These theories we call the endosymbiotic theories, lý thuyết nội cộng sinh. Symbiotic theories, where one cell engulf another cell, two distinct cells. The first cell have the nucleus. The second cell is aerobic prokaryotes. When the cells have nucleus and got the other, then they live together and one cell become the organelles of the other cell. So there is interesting facts. When the mitochondria come from the cytoplasms in the egg during the fertilization, so the mitochondria inside your cell, inside your body it just inherited from your mother. Just very few. It's not very much. We can count them from zero. It's from the sperm. So we can say that all of our mitochondria come from our mother. So this is very important for the phylogenetics and evolutionary biologies, because we can trace our source from the mitochondrial DNA. Ở đây nhá, khi mà nói về cái điều này thì có thể sao nhỉ? Cái DNA ti thể này trong việc mà nghiên cứu về chủng loại học và nghiên cứu về tiến hóa, thường thì người ta sẽ nghiên cứu về DNA ti thể và người ta sẽ lần lại nguồn gốc của loài người bằng một cái 8 mitochondria. Tổ mẫu của tất cả loài người hiện nay đó là người ta gọi là một cái 8 mitochondria. Ngoài ra khi mà nghiên cứu về xác định hài cốt liệt sĩ ấy, nhiều khi cái DNA nó đã không còn được bảo toàn, hoặc là trong gia phả của người đấy trong gia phả mà còn thực ra là người ta cũng không phải là trong gia phả. Nhất là DNA nó không được bảo toàn lắm thì người ta có thể sử dụng cái hệ gen ti thể này để phân tích cái sự di truyền theo dòng mẹ và từ đó có thể xác định lại được cái liệt sĩ. Another type of organelles I want to introduce is the chromoplast, the plant cells. Lục lạp. The chromoplasts they work to convert the light energies from the sun into sugar, so they can found in plant cells and some proteins, so that the algae. Similar to the mitochondria, they have two membranes. The outer membrane is smooth, and the inner membranes can be modified in the sac we call the thylakoid. And the thylakoid sac are interconnected to form the granular. This is structure of the protoplast. The stroma is the gel-like materials surrounding the thylakoid. So these are photosynthesis, where the purpose of the protoplast is to make sugar so they can feed the cell's machinery. Photosynthesis is process that the plant take the energy from the light and transport into the chemical, convert into the chemicals energy found in two chemicals which we call the ATP and the NADPH. And these energy-rich components will move to the stores where they can fix the carbons, fix the carbon from the carbon dioxide fixed with the water here, the H2O here to form the sugar. This photosynthesis we will discover more in the biochem subjects, not in my subjects, okay? The next about the other organelles is the vacuoles. When I introduce about the nucleus, this is the word that nucleus is the biggest organelle inside the cells, but it's not really true with the plant cell where they do have the giant vacuole. So the vacuoles is the fluid filled shaft for storage, and they can be small or absent in animal cells, but the plant cells have the large central vacuoles. In your practical lessons, the lesson number 2, you will have you have a chance to experience the large vacuoles of the plant cells. So how do we know about the organelle functions? How we can define the function of each organelle, how we can separate them from the cells and define the function of the organelles. Because you know that we can see and describe all the parts of the plant under the microscope, but we could not define and know their functions until it's become possible to relate them from the cells and separate them from one another. And these, and the process that how this thing all happens is we call them the sanctification process. So here's under the centrifugal force, in the centrifugations, the subcellular structures separate by the different mass. So when we have centrifuge by the 1000 G, we can separate the nuclear with the rate of 18,000. We can separate the mitochondria, protoplast, lysosomes. 100,000 G. We can separate the membrane, large polysomes. And when 300,000 G, it can separate the small molecules, ribosome subunits. And for the red part is the cytosomes, where I said that the cytosomes is the cytoplasm, includes the organelle. So the biochemical analysis of the isolate cell fractions can reveal what different organelle and cellular substructure do. have a video to summarize all the parts of the cells. I think we have 6 minutes more and it's not enough for these videos. Maybe you will see it at home. I will upload in the Moodle. It's quite long videos, but it's enough all the information for the lesson today. So you can see nucleus in the center. Then the rock ER surroundings, the purple part is the rock ERs, and the next, the rock ER will interconnect with the blue part here in the smooth ER, and the smooth ER also connects with the green part here is the Golgi. We have some other ties, or the RNA is floating inside cells. We can see here the green background ones is the lysosome. Minor here is like the breast, okay? Some small part here is here, cytoskeletons. Okay, these all kinds of organelles compose the cells. You can see the video as formed. And do you have any questions? I will take 5 minutes for the question for lesson today. If you have any questions after the class, you can send me to the link that I sent you. And if you have no question, the lecture will be end today.